slide preparation for karyotyping, proven protocol and troubleshooting. Welcome back to Scienceum. In this video, I'll show you how to prepare high-quality slides for Metaphase chromosome analysis, a key step in karyotyping. This protocol combines hands-on lab experience with best practices from the AGT Cytogenetics laboratory manual, so you get reliable, beautiful spreads every time. You can find more tutorials on genetic testing right here on this channel, so make sure to subscribe. Before you even touch a slide, you need to control two environmental factors: temperature, ideally between 20 degrees Celsius to 28 degrees Celsius. Humidity, best between 15% to 30%. First, you need to create a slide dropping station using a humidity and temperature monitor. You can adjust conditions using a desk lamp for drying or a humidifier for better chromosome spreading. Use only clean, grease-free microscope slides. Here's how. 1. Place 1 to 2 drops of fixative on each slide. 2. Wipe thoroughly with a non-sterile pad until you hear a squeaky sound. This means the surface is ready. 3. Check for dust, lint, or fibers. Even a tiny hair can ruin a spread. You can also pre-clean slides with ethanol and store them in dust-free containers before use. Now let's drop the cell suspension onto the slide. This step is where most errors happen. Use a glass Pasteur pipette with a soft rubber bulb. Hold the slide horizontally or at a slight tilt. Use 1 to 2 drops, spaced apart. Use ice-cold fixative in your suspension and pre-chill your slides to increase the shock, which helps chromosomes burst open. Once you drop, look for Newton's rings, concentric rings that show ideal surface tension. Drying time is a key diagnostic tool. If slide dries in under 60 seconds, humidity too low. Result: pale, patchy chromosomes with halos. Fix: use a humidifier or moisten a paper towel in a petri dish nearby. If slide dries in over 60 seconds, humidity too high. Result: dark, over-condensed chromosomes. Fix: use a lamp or warming plate to accelerate drying. Once the slide has dried, examine the metaphases under a light microscope. If the metaphases appear dark or blackened, it means the drying time was too long. If they look too pale, it means the drying time was too short. Let's look at common metaphase problems. Clumped chromosomes, suspension too dense or drops too close. Solution: dilute suspension, increase drop height, space drops. Sparse chromosomes, poor cell yield or improper fixation. Solution: concentrate the cell pellet, drop more volume. Overlapping metaphases, dropping too much or holding slide flat. Solution: drop from angle and adjust pipette wrist position. You can even try adding a drop of fixative right after cell drop. Then tilt the slide at 45 degrees and touch the tip to a newspaper to wick off excess fluid. Once the slide is dry, immediately label it with sample ID, accession number, and slide number. Place in a drying oven at 80 degrees Celsius for 17 to 18 hours to fully cure the fixative. Now your slide is ready for G-banding or other staining techniques. Slide making is a skill that improves with practice and attention to detail. Follow this protocol, monitor your environment, and troubleshoot as you go, and you'll master perfect spreads. Don't forget to like, subscribe, and check out our other videos on Scienceum for full cytogenetics workflows.